Okay, Kat, you want to switch me over?
Mm-hmm.
Okay, switch over.
Lisa, could you remind us what we're doing with these push cores?
So 5 of these will be for Paul Jensen, and he will be assaying for microbes and their biopharmaceutical potential. We're kind of hoping that there might be some weathered phosphorite minerals associated with the cores as well, and that I'll be looking at. Actually, we should let Gabe. Gabe, why don't you describe this since you're sitting right here?
For sure.
I'm going to take this back here.
So we're gonna bring this
I'll
up
take it over here.
to the ship and then section the cores from the surface down. We'll grab 1 centimeter sections, and we're gonna study which organisms are there in each section, and what organisms and bacteria, which bacteria are there, and what's their relative abundance. And we're also going to take some samples for cultivation kind of to obtain these microbes in the laboratory, grow them, and
You know we're doing 3 more, right?
so 2 different ways of studying their
Okay.
pharmaceutical potential.
We need.
Yeah. Bob didn't realize we were doing 3 more. He knows now.
Okay, thank you.
I did, I just forgot.
Okay, is that push core 5?
Yeah.
Okay, thank you. Push core 5 green tape. So that's sample NA124088.
Switch over.
This is NA124088. That was push core 5 with the green tape. Right to the left of push core 2. It's placed like right here.
Okay, well, you want me to take these all out, or
No,
do the other ones?
it'd be good to put the other ones into some undisturbed sediment. Thanks.
Okay, Al, can you zoom in? That good right there?
Sure.
zoom out. Zoom in. All right, zoom in.
So once the cores are full of sediment, they're heavy enough that they don't come out of the quiver. They don't need another rubber band.
I kind of shove them down onto the cork.
They're on a plug
Yeah.
that holds them in.
Okay.
So I give them that last little nudge to put them on the cork really good. I think that's the big deal to keep them from falling out.
Oh, cool. Just curious.
Okay, picking it up.
So push core 8 will be sample 088, is that right?
Push core 8 should be NA124.089.
Oh, 08.
Wait, who was 885?
That
Push
was
core
push core
5.
5.
What number is this?
That's 8.
Push core 8?
Yeah.
Yep.
Looks like nice, well-behaved sediment.
Yeah, so
Pretty-
far. Good cores.
Is that push core 7, the blue tape? Yep.
Yep, 7 blue.
Okay, and that should be NA124.090.
There you go. Can you zoom in just a hair? There we go. Get out of there.
stupid.
Dang it. Well, it's in there. Sorry.
Can the ones that come out of the front only go back in the front? Is there a difference? It's 6?
Well, these are going back the way they were put in. So.
Okay. Push for 6?
Correct. Push core 6. NA124091.
Can you zoom in a little bit now?
Is that push
Kylie,
core 4?
switch.
This was push core 4. Yep, purple tape.
Data logger, do you have the time for push core 5?
1700.
Thank you. Just zoned out. Thank you. Dive.
That one's not as deep.
It's number 3.
That was push core 3. Yep.
So it's a little bit shallower.
That one's good.
Push core 2?
Push core 2. NA124094.
Thank you, Bob. Record time.
Nice course.
8 cores in 20 minutes.
Sample tray in.
Yep.
They all look good.
All right.
Or 2.
Where do you guys want to go? You ready to go up towards?
Let's go rock hunting.
All right. So you want to head up slope towards WA?
Yeah.
All right.
Kylie zooming out.
Is that our first waypoint?
Well, V is officially our first waypoint, which we're very close to now. It was our landing point. Do you have any interest in going there? That was kind of on sediment, but I think we were quite happy with where we were.
Right. We want to make sure we cross over what looks like the fault line, but I assume if we head
I think
back to our original track,
yeah,
the front of the track is the same for both options.
yeah,
right?
more or less. Yeah, that's true.
So we want to be on that track.
Okay. All right, I'm looking at the pictures. Yeah, I think heading towards WA from here, we should definitely still be crossing fault line.
How far off are we from the original
From
launch site? The original bottom target.
From the original bottom target, we are 27 meters, 118 as well towards it. So we're at 295, 294, 25 meters. If you want to go there, we can go there.
Let's go there.
Okay.
Is it along the same contour? I mean, are we just, have we?
Zoom out a bit. It's within the same.
I guess we landed a little deeper than we expected. Is that right?
No, we were pretty much dead on. We expected 575 and we came in at 574. So there's a black contour band there. And then the next one is there.
And the red is our track.
That red is the 3 mile limit right now.
Oh, that's our 3 mile.
Yeah.
Okay.
Yeah.
Okay, we're not going there.
Yeah, we don't have to worry about that. That's up north. But we can go zooming back in. We can just, I mean, it's really close. We can just go there and then go up to WA.
What heading should I have?
Then we have no concern.
Yeah, I mean, I'm not too worried. We'll cross all this same feature no matter what we do. So we can head to the next waypoint.
Okay, sounds good. All right, we're going to
go across reflective area and then another sediment.
Not that one.
Don't touch it. Got it.
pilots, we're going to be heading a 076.
076. Okay, so now you got to rotate this
To change my
this
heading.
knob to the 176. Yeah.
And
076.
It's going to be about 277 meters
was it?
to
1076.
076.
But we would like to just go slow and look at the rocks so that
Okay.
if we decide to sample some, stop easily.
Sure.
Go slow. Got it.
You
Alright.
tilt down a little bit.
Okay.
Bridge nav.
And in theory,
So you're
should
just
be-
above the
should be
basket
rock.
there.
Can we get
Flip
a
it down.
20 meter step bearing
Otherwise-
076?
Yeah. It's real touchy, super touchy.
Correct, thank you.
A little more, a little more, a little more.
What's interesting, the bottom doesn't seem to be
Is that okay?
draped with
You could lateral over
siphonophores,
some.
does it?
Yeah, not too much. I haven't seen any megafauna the whole time we've been on the bottom coring.
But all these siphonophores that are hanging out above the bottom, they haven't all died and fallen onto it,
Yeah.
which you sometimes see. When that happens, it attracts scavengers, but
Is my joy gain up or down?
Yeah, you can turn that down. Turn it down like 50.
Okay.
so there's already some rocks here with some little white
0.5.
spots, which I'm going to guess are sponges.
Mm-hmm.
Do you guys want slope map up? Any backscatter, bathy? What's your preference?
Slope map is nice.
The slope map.
Okay. There's our slope. And I'm hearing the highest slopes that are showing up as red are 30 plus.
What the?
All
What do
right.
you think about these rocks, Kira? See
They look pretty angular and sedimented, so not seeing anything incredible.
That's good. You can start.
They're a little far
I
away.
think we should do a
Going in.
transect over these, or
Yeah, we could.
You guys want to transect?
I'm just looking at whether we're going to be back over sediment pretty soon. Maybe we'll wait.
Okay.
So we're just cruising then?
Cruising,
We're looking.
exploring.
I love it when
You got to move on?
We have a move on, yep. I'm going to put in another one
It's a big
right now.
rockfish type.
Bridge nav, another 20 meter
We're now
step,
shallow enough that we
076.
might start getting all those different species of rockfish.
Is it okay if I go straight?
Yeah.
Okay.
What's our oxygen level?
1% saturation.
It's pretty low. We've already got some biology. Brazilian starfish. Dead sponge skeleton.
Is that whitish? I can't.
Can you zoom in a little bit?
See a couple, a sponge.
How many little bits do you want? Half
I don't
beans,
know.
full beans.
Sorry.
Can't tell if that's actually a
don't know.
coral on the rock.
Can you get over here?
I'm trying.
Solus on
Try.
the rock top. So that's what that was. Steve says that it's a sea cucumber.
Yeah, the fake sponge.
With the tentacles out right there. It looks like, the body looks rock-like. It's colored.
It's right
Match the
center
rock.
of the rock. You can tell
This
out
is the
there.
Solus.
Zoom in.
Zoom in when you
Yeah.
want.
What is the, how does the rock look, Kara?
It
Black.
looks like
I've got more lens if you need it.
just
Okay.
basalt to me. A
Basalt.
Are you good?
Little
Yeah, I
ferromanganese
wanna go
patina,
now.
but
I want to get out in front.
not particularly fresh crust growing or those knobby, lobed textures of the phosphorites that we're hoping for. So
Video, go ahead and zoom more if you got it.
Is that a bryozoan?
Yeah,
Could be.
brittle stars all over it.
Okay, I gotta go.
I think
Come
yeah,
on.
we got to get moving. Go back to your 176 heading.
076.
076.
I'm with you.
Don't listen to me.
I knew what you meant.
Another Solus. I mean, it's funny. There are a
Can we
lot of-
kill that front porch light just for now, please?
Watch out.
Big sponge
Yeah, it's
coming
better.
up.
Thanks.
Big sponge.
More siphonophores?
Bridge nav. Another 20
Definitely
meter step.
siphonophores.
076.
The water.
Video zoom. Oops, sorry.
What are we zooming on?
I want to get that sponge. I was trying to line it up, but you came in tight.
That big, huge one?
Yeah, that one. I'm trying not to.
Don't run it over.
Sorry, I didn't do that good. Can you come on?
I'll come on.
I was trying.
Did better than I would.
It seemed like it was going to be okay, and then it didn't look that good.
Do we want to look at or collect a rock here or not?
I'm not loving any of these textures for phosphorite. But we can zoom in if we want, if we find somewhere that looks interesting, pick a couple up, rotate them around in the claw, see if they look good. We've got a,
So Kira's looking for more rounded,
Round.
bumpy, bulbous.
yeah, they don't have to be
Rocks.
spheres per se, but anything with those right
Rounded.
angles.
There's a flatfish down there.
Oh, yeah. So is this big round rock in the top left corner not?
That's really big.
Well, it's bulbous.
I like where you're going on shape. Yep.
Just making sure I understand the reference.
And then ideally they're less than 15 centimeters, so they fit
Okay.
in our inserts.
Okay.
Yeah, at this distance, it's hard to tell. So if you get to a biological area you're interested, we can
Go
zoom in
down.
on the rocks and
Okay.
check them out.
I'll step down a little bit.
Bring me a little closer to bottom. Yeah, sorry. Thank you. As long as they have enough tether to play with, yeah.
Some of these look sort of round.
Hmm?
Some of the rocks look sort of round.
Yeah.
Yeah,
See down below, some of them are more rounded.
yeah, we can stop and zoom in on anywhere and check them out closer up.
Okay. What about that guy up there?
Just like identifying a species, it's hard to tell from too far away.
See down below? Look, some of them are
Oh.
round rocks.
Yeah.
Right,
roundish.
ship's holding, so you have some time to play.
What about the one that the lasers are at the bottom of? Is that?
That's huge.
I'm just saying.
Yeah.
All right.
We need something that, like,
Round.
just about the size of the lasers, a little
Oh.
bit bigger.
Okay.
Right.
But there's
Okay.
some in along
Oh.
there, maybe.
Over there. Okay.
Yeah, let's go down and take a look.
Okay. We're gonna do this together.
Yeah, I mean, like there.
Yep.
So variety of sort of
Video zoom.
roundy, flattened ones, like that drawing.
Yeah, there's roundish
Oh.
looking rocks there.
You think here, like that, or
Oh,
this
sorry.
one?
Yeah, could be. The one to the left of the lasers right now.
Oh,
This
that
one?
in that corner. I'm coming. I'm
That
gonna-
one, right below the lasers now.
Yeah.
in here.
It doesn't have to be, like I said, not spherical, but just the edges shouldn't be sharp.
Okay. Video, come wide.
What about this one?
I'm gonna pass this to you, Bob.
Where what? No, phosphorite.
Phosphorite.
Phosphorates. That's what we're expecting to find. Or hoping. Lots of resingids on the higher, bigger rocks back there. Greg, if you're listening in the lab or wherever and you want us to do any sectioning, we can.
Okay, thanks, Lisa. I'd be interested in coarser sediments than this. I'll let you know. Thanks.
And I'll have to warn you that if you pick it up and I don't like it, I might ask you to set it back down.
You might what?
Are
If
you
we
talking
pick-
about this one?
Yeah, let's check it out.
Looks too big.
If we can zoom on
Yep.
it.
Is it too big?
Right. Too big for the bio box or too big for
Yeah,
what?
too big for the bio box.
Is it?
It's 25 centimeters long. 20.
Yeah.
The bio boxes are 15 centimeters max.
Okay.
But it'll go in diagonally.
Yeah.
Unless we put it in the big front box, but I'm not sure we want to start filling that up yet.
No.
Lose it?
Lose it.
What about this one? Is that
We can
not
pick it up.
good, not a good shape?
From above, it looks very round, bulby. The bubble view.
So that'll fit.
Can we change it, twirl it, can we zoom in a little bit? Or a lot of it? I want to see it.
You zoom in now?
Ooh, now that's some good. If we can rotate it a little. I think that's just basalt. I don't think that's phosphorite.
You do not.
No,
All right. So does that mean we don't want it, I guess?
I don't want it, thank you. This is very helpful though.
Oh, look at all the
Bromids on there?
gromids.
Yeah.
Look at all the cool things.
Oh, what
on
are those
there.
weird things? Those are weird.
Bromids, they're protists.
Ah.
Those
Those
sausage
little
links.
sausage-shaped creatures. You sure you don't want to see that?
You've got a great collection of basalts with a slight ferromanganese patina, and not a lot of phosphorites.
Okay.
I don't think it's
Well,
a
we think
phosphorite.
it's not phosphorite, so.
All right,
we'll-
lose it.
Lose
we'll
it,
lose
please.
it.
But
Got
thank
it.
you for looking. I think maybe this is not the best depositional
Okay,
environment.
so shall we keep moving?
Yeah.
Why don't we start a transect just so we get a record of
All right, sounds
what
good.
lives here? And we might stop the transect if we come across something good.
That's fine. It's your transect. You can start it and
We'll
stop it.
call this transect 1.
Transect 1. And pilots, I'm going to keep 076, but I'm going to try some 50-meter moves so we get up to speed.
Okay.
Bridge nav. Can we have a step 50 meters bearing 076?
You can just get center it up first, and then
Okay.
if I'm still on 076, right?
Not really.
Yeah, I am. I'm 076.
No, I am. You're like 60.
No, you're 60.
Uh-uh.
Gotta say she's right according to Grafana.
What?
Did I
This
change?
is I'm Argus now. I'm not her.
Okay.
Huh. Whoops. 1, 2, 3, 4, 5, 6, 76. 076.
Okay.
Well, that's going to be annoying. Go away.
I know.
Shoo.
I'm a big hand or the little hand.
All right, I'm dropping the start point. Is that okay?
Ooh,
Oh, look at the size of that sponge.
Oh my.
I bet
Wow.
Paul would want a piece.
You want to
It's
stop?
a really old thing.
You want to stop? I got to do
Video
it.
zoom?
Yeah, let's get a piece of
Get
it.
at least
Bridge
a
nav,
photo.
hold position.
And a really good
Well, I set it
photo
up so
before
nice.
we do anything.
Come on. What's about the pirouette?
I'm coming out all the way.
That has to be an ancient
you.
sponge. Beautiful.
Wow.
You got some captures?
yeah, I'm getting some good pictures.
On just a giant basalt spire. They like being high up. Reminds me of cats.
Cats.
They like vantage points.
Ospreys also.
Yeah. That is incredible. Any
Oh no, I'm a little scared that we're gonna hurt it.
I'm not driving, don't worry.
No, I meant when we if we section a piece of it. Paul, are you there? Can you speak up on whether you want this or not?
What's the texture like?
Spongy.
Is it a glass sponge or is it
Right.
like bass sponge?
I am not sure. If anybody's on watching this
That
and can
could
tell
be a
us what sponge this is, please do.
You want to zoom in some more, Al?
There's our friend.
So, Caitlin, maybe you could find Paul and ask him if he would like a piece of this sponge or not.
Wow.
before
Jesus.
we go in there.
Sorry about that.
And I was saying basalt, but there seems to be some kind of layered textures in that rock. Could be sedimentary.
Could you zoom it in a little bit more?
Why don't I land and then we can zoom way in on it?
Maybe the sponge eats
I think you'd have to come down there, Kelly.
siphonophores.
I'm getting kind of yanked.
Steve said
Okay.
there's some primnoid octocorals at the base of the sponge also.
Can you zoom out, Al? really strange rock formation
It's pretty cool.
there.
I like it.
We're told it's a basalt spire.
Well, now that I look, though, that you see there's these
Layering.
bedded layering, that's probably more indicative of sedimentary rock. So it's,
Looks like a horn.
yeah.
It's just uplifted into that position.
Yeah, either fell that way or got squished that way.
You can zoom in on Argus if you'd like. Try to center that for you.
So we're not hearing anything from either person in the Jensen lab. What do you think, Gabe? Do you guys want this?
I think so. Yeah.
Okay. So if we can find a little lobe to suction that's not going to damage the big thing or push it off the rock, that would be good.
It's a cool Argus shot too.
You want the tether out of the view? Oh,
How tall do we think this sponge is? Like a meter?
okay. Okay.
Okay, Al, can you zoom in?
Ooh,
Whoa.
look
Wow.
at that.
So cool.
See all the channels? Looks like the holes are plugged with sediment, doesn't it? You know,
Some shrimp on there too.
blue shrimps. Oh, there's polychaete, I think, right there. Another shrimp.
I think that's a hydroid, the feathery thing coming out.
Hydroid?
Hydroid? Upper right.
Yep, right. You can't see me drawing.
They can actually put that up on the over the satellite.
I'm just
Yeah, I
thinking
can put that on
about
channel 3.
whether one of the lower lobes, you know, lower bits of the sponge.
I
where we've
assume
been-
that's all the way in, right? Al?
Yep. And telestrator is up on channel 3 on the quad, bottom left.
So if you draw on it, other people can see.
Okay. Well, this is what Greg was saying is a hydroid.
So, all right, up a little bit, try to expose it.
There's a shrimp. But Bob, I think for sampling, if we could sample a lower part.
Yep. Okay.
Good.
Okay, Al, zoom out.
You want to come on all the way?
Yeah.
Are you ready for sample? Yeah.
Have something off the bottom
Yep.
vicinity.
I don't have a really glorious perch here. I'm sort of precariously balanced, but we'll see how it goes. So are we gonna try and, we could grab and slurp?
Yeah, I think we'd like to put it in this one of the course slurp section. That's
Okay.
4 through
Al,
7.
you wanna set that up?
Do you mean there or down lower on the rock?
Lower a little bit. Can we reach
That's-
the
well,
lower
that's,
lobe?
That's kind of harder to grab.
But whatever you can reach is fine, but I'm just
can-
hoping to
can
do
gr-
the least amount of disturbance.
Yeah, I was just gonna grab the corner of it up here.
But do you think that will dislodge it? 'Cause
No, I don't. I think I can
Think
grab
so?
just a little handful of it.
And can I have some porch lights, please, when you get a chance?
So Steve
Riley?
is
Yeah.
telling us that this is not a known sponge,
Thank you.
Oh, cool.
and it's not in any of the guides that we have on board, which could be a new sponge, I suppose.
Hey, Aaron, that says 7, right?
I'm worried that that's a
Yes. it
prime
does.
anchor for the sponge. I guess if you leave the base
Yeah,
attached to the rock and
I'm
you
just
just
gonna
take
grab a wee bit,
okay.
a bit off the top here. How much do you need?
Handful,
Cloth.
a clawful. Sorry.
All right, let's not forget to log the sample too.
This can be, where, do we know which section it's going to go in?
7.
7. Just worry that it's part of the anchor system.
Oh, I think it attached all the way along the bottom edge.
We'll never know what hit it. All right, zoom out now.
Okay. Where are we putting this?
In the slurp.
Slurp 7.
Oh, okay.
We hope. Let's see if we get it.
Paul is saying more, if we can get more.
Right.
Okay.
Well, I think little bits at a time is better here. For
Ready to turn suction on?
Yep.
Yep. Okay. 100%?
Yeah.
Watch the sample jar, make sure it comes in. It's at 100%.
Right.
Doesn't seem to be sucking, does it?
Oh, I think it's just
Going in pieces.
stiffer than it appears. First glance.
So
Get
it
in
doesn't
there.
fit. Should we put it in one of the compartments?
It is pretty stiff.
Okay. How hard would it be to set it in one of the spaces in the starboard bio box?
The starboard bio box is like anything that's sort of floaty, which I think this is very hard to keep in the box because the wash over the box, you got to roll it in and out.
Maybe on the spaces in between.
That's what he's saying.
That's the
Anything in that box. I don't know that that sponge floats. It fell. The piece you dropped fell without floating. But it looks like we're not going to fit it in this section. Did a piece go in already?
Yeah.
Yeah, I think a little bit of it.
Suction off.
I mean, if
Did anything go in the jar?
Just fragments.
Yeah, nothing obvious.
not enough for them to work with.
All right, turn it off.
Suction's off.
Okay, so Caitlin and Paul, are you okay if we donate one of the front compartments to this sponge and we can put a lot of other animals in there with it as well? You okay with that?
Yes, that sounds great. Let's start doing that.
Let's just put it in one of the front bio box compartments.
Okay. Zoom out.
And we'll mark that as one that we'll continue adding it. And Lisa, you're correct. It looked like this will sink in there, so that's what we're hoping for.
Say that again?
It looks like your observation
yeah,
that it's the other
you gotta
piece
do
off.
it without knocking me off.
Hopefully it'll sink on into that box.
Stupid cable.
Yeah, that doesn't look so buoyant.
Hate this.
Follow the wires?
Exactly.
Yeah.
Yeah, so if we could get this
Okay.
piece and perhaps another piece,
Someone else.
it'll sink right in there.
Careful, careful, careful. You're gonna knock me off. So just hold it there.
Okay.
I'm
We're
I'm
right
pretty
up against the rock.
precariously.
Positioned.
Going for red 5.
Red 5.
What's the sample number called?
NA124.095.
Are we marking both as samples since something went in the Niskin? Or not the Niskin, I'm sorry, the slurp jar.
Are we going to count that?
I think we'll give it the same sample number.
Okay.
Like another.
Okay. We zoom in again.
Is the coral of interest from this rock to anybody? Greg or Paul? Primnoid. Oh, not too much.
Oh, too much.
Oh, please.
Attention.
Pretty small to grab. Not sure how you'd really sample it.
The coral?
Yeah, the coral. Does Steve know what it is?
I think Steve is saying it's Peristanella ramosa.
The sponge?
No, the coral.
Coral. Well, there's a really large piece of a very old sponge. Should be
Let
enough
me
for
know when
everybody.
you want the tool tray in.
Fantastic. That was beautiful. Thanks so much.
Okay.
Is that okay?
Yep.
All right,
Okay.
coming in.
Okay. What are we trying to sample now?
Nothing. I think we've decided not to take it, to continue our transect.
That wasn't that big a piece of the sponge.
It's okay. You're okay. That was a nice sample. Thank you.
It was
good
a great
sample.
sample. He's going to be fine.
It's just such a pretty sponge.
Thank you.
I love that Argus view.
Isn't it
So
cool?
dramatic.
Somebody take a capture of that.
Yeah, I've got some nice pictures of Argus.
It's going to be my computer background.
You want to get set up first, or should I start ship move?
I'll get set up first.
Okay.
Can you come wide on Argus, please? Thank you. Do some tether management.
We 076 again?
Yep. I think so. Let me just check where you are now. Where are...
080.
And the sponge, can you just give us a clue about what we would call the substrate that sponge was living on? Sedimentary
Yeah,
spire?
sure.
Or
Sedimentary boulder. Maybe spire is too specific.
Come down on my delta. Okay, you're pulling me.
Sorry.
We have
Okay.
not collected any rocks yet, Caitlin.
So it's interesting. We're in a really pretty flat area,
All lined up. All right.
which on the slope
Bridge
map
now.
we would have perhaps considered to be sedimented. But
Can we get a 50 meter step bearing 080?
Yeah, it basically looks like a talus pile from the slope above. I think it would be okay to do a are we doing a transect?
we are. We're continuing, right?
We never started.
We never started?
And we saw the sponge.
We didn't go anywhere.
We
like,
saw a giant rock.
yeah.
Okay, so starting now.
So we're doing transect, so no zooming and no deviating,
stop.
just drive straight line.
Okay.
Just keep it.
We can, I mean, it's okay to stop and look at things.
But we truly hadn't even had a chance to drop a point, so we didn't start the last one. But yeah, once we're started, you guys, if you want to keep calling it, that's fine.
So it's not an official transect?
Yes, please. I think we can
So
get
no,
through this.
no
boulder
zooming
pile.
and
Well,
looking.
that's up to them. So Lisa, what are your transect rules? Zoom okay, stopping okay?
Well, we want to keep the same view, but then if we stop and zoom, we just pick up the transect
Okay.
where then we go back to being a meter off the bottom or so. We'd like to stay within a meter. We can.
So again, if we're going up slope, the rear of the vehicle is where the altitude's measured. So the front's lower than the back. But we have to look over the top of the tube cores. So if you want to look down, we're going to have the tube cores in the view.
So does that mean the back should be 1.5 meters off the bottom, or
Well,
where the
there's-
altimeter
I mean,
is?
You could try and do the trick, but it's kind of hard to figure out if you don't know the angle of the slope. I'm just saying we're closer in the front than what the altitude reading is
Okay.
because we're going up slope. So
I'm not seeing the altitude reading anyway.
It's 1.8 right now, 1.7.
See.
It's on the Herc screen below.
so if you look
Right
at
here,
this,
Lisa.
if you look at this, the bubble cam view, it shows how high we are at the front of the vehicle.
I see.
So we have to keep that, otherwise we're bouncing off the rocks. So, see how close it is?
Yep, looks very close.
Yeah. So despite the altitude being 1.6, the front of the vehicle is almost scraping the ground. And then when we come up on things, we're going to have to step over them, right? So, but we're looking up a bit because of the tube cores. So if you want to like look down and get a better view of the bottom, you have to deal with the tube cores in the picture. So which would you rather
I think
do?
we're okay the way we are.
Okay.
Okay.
It's just it's all gonna be kind of greenish and far away if we're doing it like this. Is it okay the way it is?
Well, you can't really drive if we zoom in anymore, right? Because you need to be able to see where we're going.
No, we could probably do it because we're kind of trying to stay off the bottom with this other view and not knock over sponges and whatever, right?
Yeah.
Well, we could.
Some asteroids.
You want to tilt down a little bit?
Towards me?
No.
Oh,
gosh.
The other way.
Like an airplane.
The other down.
So, yeah, see, we can't really look down very well because of all the... Is the camera all the way out?
Of course.
No. Yeah, it's all the way out. So, yeah, there's that view. Or there's above it and zoomed in. You want to zoom in a little bit, Al? Does that help any?
Maybe we just pan up a slight bit so the cores are more towards the bottom.
Because I usually do the
like
bubble.
that. Let's leave it like that.
Okay.
Which light is off, right?
What's the distance to
Does
WA?
look pretty
Aaron?
green.
How's that?
It's okay.
Okay.
What are those distances you're getting, Aaron?
I'm just estimating
I'm
100
watching.
meters.
Oh.
So that, do you see where my mouse is?
Yeah.
That's about 100 meters.
Yay. 100 meters till the face. I'm excited. Let's do it.
We can go 0.3 if it's... There aren't too many big boulders to run into.
How far are we gonna go?
We still have to go about, where's my mouse? 78 meters.
Oh,
I can ask him to bring it up to 0.5. I think it's at 0.3.
yeah.
Okay. Bridge nav. We have another 50 meter step bearing 080, and if it hasn't been set already, can we increase the ship speed to 0.5 knots?
Oh, there's one of those
Yep, thank you, George.
Sacco calyx. So I'm trying to think about where the fault line is. Could you put up the bathymetry just briefly when you get a chance?
I think it's at the base.
Is it through here?
I think it's like right along the base of the slope, I think.
Yeah.
I wish I had flater mouse up here. I hate like looking at things in 2D.
Okay,
I'm
so
spoiled.
right at the end of the transect.
Yeah, I'm pretty sure, because I think if we like zoom out and you're looking at the dive plan, I'm pretty sure it was that kind of that line where they got that really steep scarp, seemed to line up with the what we thought was a fault, and that would be right along there. That looks like it to me, at least.
It looks like there's a Panicia sea urchin down there. I mean, a sea cucumber, also some sea urchin.
So it looks like we're going slightly downslope here.
That's probably right. There's a little dip and not a trench, but there's the fault line is like a little depression.
Yeah, and it did actually look like it had sediment in it, because there was a dark line when we were looking at the backscatter, and we're like, oh, I wonder why like that.
So this is probably the start of sediment.